Okay, we're back in repro2, reproduction 2. Starting with the anatomy of the perineum. I want to put the perineum up here, but this is the similarities and differences. The perineum is the space between your anus and the orifice of your urethra. It's obviously a lot longer in males than it is in females. Remember, male and female external genitalia is homologous tissue, along with the gonads. That is, it's from the same fetal tissue. So the glans penis is homologous to the clitoris. The labia minora is homologous to the shaft of the penis. The labia major is homologous to the scrotum. Oh wait, the current slide. And there's the perineum. This is on top of page 1106. You have obviously male-female external genitals. The area within the labia majora in females is called the vestibule. There's a anal triangle from the two ischiopuberosums to the, posterior tip of the anus, and then from the ischiotuberoses up to the pubic symphysis. It's called the urinal triangle, and you can see a lot of these structures of homologous. Also, some of these muscles, bundles, ischiocavernosus muscle, bulbosus muscle, perineal body, perineal muscle, they're all homologous. In males, they help with ejaculation, and some of these muscles. In females, they help with what's called the orgasmic platform, how the vagina can grasp the penis during coitus. Okay. Gametogenesis. This is the making of gametes, and it's also called meiosis. So remember what diploid and haploid means. Diploid is the total number of chromosomes you have in normal, what is called somatic cells, cells that are not involved with reproduction. And that's 46. That number is 46 in people. Haploid are gametes, half the number, 23. And gametes are the only haploid cells that human beings make. Okay. So we have 23 pairs of homologous chromosomes. 22 of those pairs are called autosomes. They're not in with gender determination. And then we have the sex chromosomes. And two types, X and Y. Okay. Meiosis. Differences from mitosis. Mitosis is an asexual process. It makes two cells from one cell, and those two cells are genetically identical to the mother cell. Right? So it starts off with a diploid cell, makes two identical diploid daughter cells. Okay. Has 4 phases. It's a one-stage process, 4 phases. DNA replication in meiosis is going to be 2 stages with 4 phases each, and it's going to make, at least in males, it makes 4 functional gametes, right? And it's called a reduction division because it makes haploid cells. DNA has to be replicated beforehand in what's called the S phase of the cell cycle. And then when you make double chromosomes or replicated chromosomes from single chromosomes. And therefore you have in a replicated chromosome, it's like an H, and it has 2 what are called sister chromatids because they're genetically identical to each other. Now the chromatids from one homologous chromosome to the other homologous chromosome, they're called non-sister chromatids because they can be genetically different from each other. there's going to be different alleles at some of those loci between non-sister chromatids. All the alleles are identical in the loci and sister chromatids. They could be different. They could be the same, but they could be different in non-sister. And during meiosis, do you not only divide the number of chromosomes in two, you scramble them in a process called crossing over or recombination. How parts of homologous chromosomes cross over, break off, and give each other pieces. So you really scramble your chromosomes. You are not going to give your kids the same chromosomes that your parents, or same combination of alleles on chromosomes that your parents gave you. You're going to scramble. Okay, reduction, as I said, reduction division divides the number of chromosomes in two. Separation of tetrads. Tetrads are two homologous chromosomes next to each other. In the first phase of meiosis, called prophase 1, tetrads form. Homologous chromosomes find each other. How they do that, I don't know, but they find each other and they go right next to each other. That's called a tetrad. And then they cross over, and then they get pulled in the middle of the cell in what's called metaphase 1. And then, and which one gets pulled in what direction? What within the father's chromosome gets put to the left, and the mother's chromosome to the right, or vice versa. It's completely random. It's called independent assortment. As a matter of fact, you can line up 23 pairs of chromosomes 8 million different ways in the middle of the cell in metaphase 1. In anaphase 1, these tetrads separate, and you already form haploid cells. That's a big difference in mitosis. In mitosis, every chromosome lines up straight like this, and then the centromere splits, and the sister chromatids go their separate ways. Here in metaphase, in meiosis 1, homologous pairs form up, tetrads, and those separate. And then in meiosis 2, you separate sister chromatids. Then you line them up, and you break centromeres, and you separate sister chromatids. So meiosis 2 is really exactly the same as mitosis. It's just done with the haploid number of chromosomes. So here is a diagram of meiosis 1. This is on pages 1108 and 1109. Prophase 1, homologous chromosomes find each other, the nuclear membrane disintegrates, spindle fibers come along attached to centromeres. Crossing over occurs between non-sister chromatids. Then in metaphase 1, they line up in the middle of the cell. Interphase 1, they go to separate cells. Telophase 1, they become different cells. Right? And you already have haploid cells, but you have a haploid cell with a haploid number of replicated chromosomes. And gametes have the haploid number of single chromosomes. So you have to go through meiosis 2 to determine the single replicated chromosomes into single chromosomes. Okay, and there's meiosis 2. And it takes place in two cells, because now you have two cells that are formed after meiosis 1. And then you attach the spindle fibers to them in the prophase and line up metaphase 2. The centromere split and the sister chromatids go apart now. In anaphase 2, and then we have a pinching off of making 2 more cells in telophase 2. And now we have 4 haploid cells. And remember that pinching into 2 cells during telophase is called cytokinesis. Okay, gender differences in Then differences in meiosis. Timing is one of them. When does meiosis start in males? Puberty. When does meiosis start in females? When they're fetuses. And they already go through, they start prophase 1 and they get arrested in prophase 1. So when a little baby girl is born, her oogonia, I've already started to make gametes. And they're arrested in prophase 1, and then they won't restart meiosis until puberty. So there's a difference in time. Also, difference in results. When meiosis is complete in males, 4 functional gametes. In females, 1 functional gamete, and what are called 2 polar bodies. The first polar body, that is 1 to 2 cells that are a result of meiosis 1, disintegrates. It becomes this non-functional thing called a polar body. Then, and then the interesting thing also, when does meiosis end? Well, in an individual oogonia that becomes a oocyte, if a woman's egg, oocyte, is not fertilized, so what is sent out of the ovary at ovulation, it's a multicellular thing, but it's called a secondary oocyte. One of the cells is called secondary oocyte. It is a haploid cell with replicated chromosomes. It is arrested in metaphase 2, and it lasts about 48 hours. If it's not fertilized in 48 hours, it will disintegrate and never even finish meiosis. Because what's the point? If it's fertilized by a sperm, then it finishes meiosis. Goes to finishes metaphase 2, interphase 2, telophase 2, makes a second polar body, which is a haploid cell with a single number of haploid chromosomes, and it disintegrates, or it degenerates into what's called a second polar body. It's kicked out. And then the 2 sets of chromosomes from mom and dad come together, and you have a zygote. Okay, disjunction and non-disjunction. Disjunction happens in anaphase 1 or 2 of meiosis. In anaphase 1, tetrads are supposed to separate, but sometimes they don't. Both homologous chromosomes go into 1 cell. So 1 cell will get an extra chromosome. The other cell will be missing a chromosome. So this is called non-disjunction. It doesn't happen. And so this is going to end up with what's called an aneuploidy, or an abnormal number of chromosomes. And this is talked about on page 1110. And there are 2 types of aneuploidies, what are called monosomies, which are a baby that's missing a chromosome. And this is almost always lethal. There's only 1 non-lethal monosome. It's called Turner syndrome. It happens in the sex chromosomes when a little baby girl is born with without the second X chromosome. She only has 1 X chromosome. And sometimes there's this problem. Sometimes this person looks completely normal. Sometimes there's some problems. Trisomy is the other thing, too many chromosomes, 1 too many. And generally you think, well, why, I can see why missing a chromosome might be, a bad thing, but why is too many? Well, it is. And so this person has 47 chromosomes rather than 46. The monosomy has 45 rather than 46. And they are all lethal. Really, trisomy, and they call trisomy after the number of the pair that undergoes non-disjunction. So trisomy 1, 2, 3, all the way down to trisomy 18 are all lethal. Sometimes a little, if we're born with trisomy 18, it will live a little short while, but not too long. The most common trisomy for some reason is trisomy 21, which is also known as Down syndrome. That's a little blurred, clear view on 1110. It's Down syndrome. And then there are a bunch of, and that's an autosomal trisomy. There's several trisomies of the sex chromosomes. There's something called a triple X female with 3 X chromosomes, and there is no problem with that's a metafemale. That's a metafemale. There are no problems with that, because it's an interesting thing that in females, one of the X chromosomes shrivels up in all of their cells. And so females have 2 X chromosomes, but their cells only use one of them. One of them shrivels up and becomes a non-functional structure called a barr, B-A-R-R. And in half the cells, dad's X chromosome becomes the barr. In the other 50% of cells, mom's becomes the barr. It's random. So females are what's known as natural mosaics. That is 2 different genetic combinations in the same individual. Because remember, the 2 X chromosomes have the same genes, but they could have different alleles. So in some cells, those cells are using dad's allele on the X chromosome. In other cells are using mom's allele on her X chromosome. Okay, then there's what's called Klinefelter syndrome. 2 X's and a Y. And these are males. They usually have underdeveloped genitals, and they have female secondary sexual characteristics, that is, they grow breasts and so on. And they usually go through, life as males. And then there's Jacob's syndrome, or the XYY males, 2 Y chromosomes. Of course, this happens in male. This must have happened in male meiosis, where you have an XYY, because trisomies usually happen in female meiosis. And why is that? It's because tetrads are formed in females when a female is a fetus. They're formed, and they're held together for 12 years until puberty. It's under some mysterious process. Several of those cells are meiosis turned on in several of those cells. And they go through some like a competition, I think, for FSH receptors. And one of them wins the competition, the others degenerate, and that one becomes egg and lump. But here's the thing, the longer these homologous chromosomes are held together, the more likely they are not to separate in anaphase 1 or 2 like they're supposed to. Some of the glue gets stronger or something, particularly after age 35. Why is this? Nobody knows, but the risk for an aneuploidy is really low for women from puberty up to age 35. And then all of a sudden, age 35, it just goes like exponential. So any woman that's over 35 and is pregnant, their obstetrician is almost always going to suggest amniocentesis, just to see if, see how many chromosomes there are. And then you can make a decision on what you'd like to do. But XYY, that had to have happened in males because females don't have a Y, so how could they have a non-disjunction between Ys? So anyway, but here, so those are called chromosome mutations, when you have changes in the whole chromosome, aneuploids, more common in females. But gene mutations are mutations in individual genes. And this happens during DNA replication. Whenever you replicate your chromosomes, you make errors. You're going to make errors, mutations. And so here's why they're more common in males. How many times does a female replicate her chromosomes and make a gametes? I don't know, a couple of times. If she's born with all the oogonia she's ever going to have, those chromosomes have already been replicated. And so they're only replicated a few times. And they're not ever going to be replicated again. So you can't make mutations if you don't replicate. How many times do males replicate their chromosomes? Gazillions of times, because every time they make sperm from a spermatogonium, they're replicating more chromosomes. So that's where most gene mutations come from, males, because how often they replicate their chromosomes. Okay, male anatomy, scrotum. What is its purpose anyway? Why do we need a scrotum? The whole testes. Well, yeah, but why are testes held outside the body, in a sac that's outside the body? Do all male animals have scrotums? No, they don't. There's several animals. Pigs, for example, no scrotum. Whales, no scrotum. That would be a really big scrotum. What has happened there was something happened, some mutation happened in spermatogenesis, making sperm that it doesn't work at body temperature. If you try to make sperm at body temperature, you make lots of defective sperm. So they have to be held outside the body, held about 2 degrees below body temperature. And then you make good sperm. So that's the purpose of having a scrotum and having testes outside your body cavity, because it's a little cool. Because there's a counter-current heat exchange, that hot blood coming into the scrotum comes by cool blood coming out of the scrotum, and the heat leaves from the arteries coming in. Heat goes to the veins that are leaving out, and it keeps those testes a little cooler. Matter of fact, there are some muscles. There's a dartos muscle that covers the testes. There's a pampiniform plexus, which is the series of arteries and veins that are right next to each other when this counter-current exchange occurs. And then there's a cremaster muscle that will pull testes up or relax and let them hang lower according to the temperature, to the external temperature. If it's really, really cold, the cremaster muscle will pull the testes up close to the bodies that are warmer. It's a really hot day, and relax and let them swing farther away just to maintain that 2 degrees below body temperature. And there, obviously, are male genitals. All right. And we'll talk about in lab about the erectile tissue, the corpus, what's called corpus cavernosum, corpus spongiosum, and we'll talk about the different ducts leading from the testes to the scrotum, the vas deferens or ductus deferens that brings sperm through the seminal vesicles, through the prostate, and it's called the ejaculatory duct, and it joins the urethra in the prostate, in the prostatic urethra. Then the urethra goes to the urogenital diaphragm. There's a bulbourethral gland there also, and we'll be talking about what these glands, the secretions these glands later on, the secretions of the seminal vesicles, the secretions of the prostate, the secretions of the bulbourethral gland, and make up they make up semen. And so secretions in semen are made by those glands. The cells in semen made by the testes and go through the vas deferens. And then the spongy urethra goes to the corpus spongiosum and enlarges corpus spongiosum, enlarges at the end of the penis, with the gland speeds. And the epididymis is an area right next to the testis that sort of stores sperm. Okay. So the testes are a series of tubules, called seminiferous tubules. And inside these tubules, spermatogenesis is going on, making sperm. There are cells outside between the tubules called Leydig cells or interstitial cells. They are cells that actually secrete testosterone. There's a germinal epithelium around the outside of these tubes, and as you move in, and that's where the spermatogonia are. There are sustentacular cells, or also called Sertoli cells, that support and surround the spermatogonia. There's a blood testis, and they're part of what's called the blood testis barrier. You have to separate testes from your immune system in the blood because they're different genetic combinations, and they might actually attack sperm. It's a mediate testis, a mediastinum testis. This is a series of tubes that come from the seminiferous tubules that coalesce into the epididymis. There are nerves, both sensory and autonomic nerves, in the testis. And there's a cross-section of the anatomy of the testis, and let's go to the mediastinum testis, and the efferent ductules becoming the body of the epididymis. And we have connective tissue on the outside of the testis, and there's the ductus deferens leading up to the spermatic cord. The spermatic cord goes through the inguinal canal into man's abdomen and heading on to the seminal vesicle and to the prostate, to the ejaculatory duct. And there's a cross section showing interstitial cells, the seminiferous tubule, the tubule lumen, the germ cells, cross section of a seminiferous tubule. So the efferent ductules, the seminiferous ductules become the efferent ductules and the mediate testis. They lead into the epididymis, where sperm can be stored for 40 to 60 days. The ductus or vas deferens, when it gets into the seminal vesicle, it gets a little larger, called the ampulla, joins with the ducts of seminal vesicles that make a fluid that is put into the ejaculatory duct. Ejaculatory duct passes to the urethra and passes to the prostate and unites with the urethra, and there's a bunch of little holes in the urethra, in the prostate, where the prostatic fluid enters the urethra, and that's where semen builds up before ejaculation. Okay, accessory. So seminal vesicles provide 60% of semen, prostates provide 30% of semen, so that means 10% is sperm, right? And we'll look at what's in those in those fluids in a moment. The bulbourethral gland, also known as Cowper's gland, this is a pre-ejaculatory fluid. And it's done for two reasons. This is when it may get sexually excited, makes this mucousy, clear liquid that does two things. One, it lubricates the urethra. A lot of people think it's lubrication for sexual intercourse, and actually it would help in that, but not use that much to do a lot of good in that regard. Usual lubrication for sexual intercourse is mostly provided for by the female. But what it does is it does lubricate the urethra. And it also buffers the urethra. But because, remember, in male urethra, at least across the spongy urethra, does double duty. What was the last liquid to pass through this urethra? Is urine, which is usually acidic, right? And the sperm don't like acids. They don't like acid. So there's a buffer in the bulbourethral fluid that will neutralize acid. And also, these little cells are fragile little cells, and they can be expelled from the penis with real force, and they can hit the sides of the urethra and really get damaged. So this pre-ejaculatory fluid is really mucousy, so it provides a nice soft mucus layer around the urethra. So when the sperm gets shot out during ejaculation, they're not injured. So penis directionality, ventral dorsal. Here's this strange thing. We're primates, and primates have a pendulous penis. Lots of other mammals, the penis is attached to the body cavity. So you can see what part is dorsal and what part is ventral. But in a pendulous penis, actually the dorsal part becomes the ventral part, and vice versa. That's not really important, but it is interesting that the directionality is reversed because it's pendulous. Okay, shaft of the penis, we talked about that. Glands. The glands means the expanded head of the penis. It's usually covered with a prepuce or foreskin. When little boys are born, they're all born with foreskins. They're often removed with circumcision. But if they're not, then you have to worry about something called smegma. There are little glands on the glans penis, and the base of the glans penis to make this fatty substance called smegma, and it's really food for bacteria and everything. And so little boys who are not circumcised should be taught to roll back their foreskin when taking a shower or bath and clean that area up. It's maybe one of the reasons why men circumcised have a slightly lower risk of penile cancer than men that are not circumcised. Okay, the penis, remember, has erectile tissues. So do females. We're going to get to that. But there's one called the corpus spongiosum, and that's surrounded by, and the base of the penis is called the bulb, and it's surrounded by a bulbospongiosis muscle, which is misspelled on the slide. Anyway, and then it's corpus cavernosum. The base of it has two pointy parts. The base of the penis is called the crux, and it's surrounded by the ischiocavernosus muscle. And these things squeeze the penis during ejaculation and help to expel semen during ejaculation. Okay, puberty and climacteric. Definition is adolescence and puberty. So adolescence is before puberty. Puberty is when we can actually make gametes and we can make babies at puberty. Climacteric is the end of making babies. And it happens in females in menopause, it really doesn't happen in males. Okay, so there's a slide of the penis and the two erectile tissues with the bulb and with the crux. And cross sections of the ductus deferens, the prostate gland, and seminiferous tubules. So remember, there's and this is all really hormonally controlled. Remember, there's a hypothalamus, a pituitary testis axis. And this, okay, the slide, the figure I just showed you is on page 1137. This hypothalamus, on page 1133, the hormone regulation is spermatogenesis and estrogen production. So GnRH, gonadotrophin releasing hormone, released by hypothalamus. They travel through the apophyseal portal system to be released into the anterior pituitary. The anterior pituitary or adrenal hypophysis makes FSH and LH, follicle stimulating hormone, and luteinizing hormone. These travel down to the testis, and there's two testicular secretions of testosterone from the interstitial cells and something called androgen binding protein from the sustentacular cells. And these stimulate spermatogenesis. And then the presence of testosterone is going to feed back upon the hypothalamus and interpituitary to turn off FSH and LH. And inhibin is going to do the same thing. It's an inhibitory hormone that's going to turn off the production of FSH and LH. And so this is figure 2817 on top of page 1133. GRH secreted by the hypothalamus stimulates the interpituitary to secrete FSH and LH. LH stimulates interstitial cells to secrete testosterone. FSH stimulates sustentacular cells to secrete androgen binding protein, which keeps testosterone levels high in the testes. So here's the thing, testosterone is needed in higher concentrations in the testes to support spermatogenesis, higher than it ever gets in the blood. So what androgen binding protein does is it binds the protein and makes its concentration really high in the testis, high enough to sustain spermatogenesis. Testosterone stimulates spermatogenesis, but inhibits GRNH secretion and reduces the anterior pituitary sensitivity to GRNH. So there's a negative feedback of testosterone back to the hypothalamus and anterior pituitary to turn off GnRH and therefore FSH and LH. Rising sperm count levels cause sustentacular cells to secrete inhibin, which further inhibits FSH secretion. So here's the thing. So inhibin just inhibits FSH, which stimulates androgen binding protein. So inhibin is going to turn off FSH. Androgen binding protein will be made, and then testosterone levels in the sperm and the testes will drop. But they won't drop anywhere else in the body. They'll stay in the same level in the blood because testosterone does other things. One of which is maintains secondary sexual characteristics. So if you want to keep testosterone high at the correct level in the rest of the body, but want to lower it in the testes because you already have enough sperm, then you secrete inhibin, which inhibits FSH and stops androgen binding protein. So testosterone stimulates development of secondary sexual characteristics and libido, which is sexual desire. Testosterone is responsible for sexual desire. It stimulates sexual desire. Okay, myoclimacteric or andropause doesn't really exist. Men make babies. When do women stop making eggs? Menopause, somewhere around from late 40s to mid 50s. The reproductive life ends. When does a male stop making sperm? When he's dead. That's when he stops. There is no termination. Why is that? Lots of speculation from biologists and people about why this is, why women have an end to their reproductive life in menopause and men don't. Nobody knows, but that's the way it is. Okay. Spermatogenesis, male meiosis, two-stage process, remember, formation, we've already gone over this. Formation of tetrads, crossing over, independent sorting. And two types of spermatogonia, we talk about that. One type of spermatogonia just goes through mitosis and makes more spermatogonia. So men make spermatogonia their whole life. Females don't. They made all the oogonia they were going to make as fetuses. So they don't make new oogonia, but men make more spermatogonia. And we don't have to go through what the difference between primary and secondary. Spermatocytes are spermatids in what's called spermiogenesis, which is the last part of making sperm. It has an unfortunate name because it sounds just like spermatogenesis, but it's just the last stage of spermatogenesis. Let me show you that on a figure here. And this is, this is on page 1134, and this is just a spermatogenesis. And you're making, you're going through meiosis, and you're making these things called spermatids, and spermatids become fully functional sperm. Remember, sperm have a sperm head with 23 single chromosomes. They have a midpiece that holds mitochondria that fuel the flagella that allows them to move. And they have what's called an acrosome. On top of the head is an acrosome that's filled with digestive enzymes. And we're going to learn what that's for later on, particularly the next chapter. We'll talk a little bit about fertilization, what's called the acrosomal reaction. And remember, the very last part of making sperms is called spermiogenesis, not spermatogenesis. Be careful about that. Okay, so as I mentioned, spermatocyte, a head with acrosome, midpiece with mitochondria, a flagella. Okay, semen. Usually, the volume of an ejaculate is 2 to 5 milliliters. As I said before, 10% sperm, 3% prostatic fluid, 60% from seminal vesicles. It's going to be a little bit of bulbourethral fluid in there, because it goes through the urethra after the bulbourethral glands that are secreted there. So it's going to have a little bulbourethral fluid also. Count 50 to 100 million sperm per milliliter. So the typical ejaculate is going to have hundreds of millions of sperm. And one of the major causes of male infertility is low sperm count. You got to have at least 100 million, or you're not going to be making a baby. Why so many? I'm going to hold off on that until a little bit in the next chapter. We talk about more about sperm's journey after sexual intercourse, and what they have to do, and why so many sperm are needed to let one sperm get inside the oocyte. So here's semen. The prostate gland secretes what's called a clotting enzyme, what's called prostate-specific antigen, seminal plasmin, citric acid, calcium, and phosphate. The seminal vesicles secrete a fluid that has fructose in it, sugar, and this feeds the mitochondria and allows the ATP to be made for the flagellum to operate. Prostaglandins, it is thought that prostaglandins might actually constrict, cause the constriction of female smooth muscle to assist the sperm. And what happens is the seminal plasmin clots. There's a clotting. It's a clotting enzyme. When sperm is ejaculated, it forms a clot. It's thought maybe that it helps it stay within the female reproductive tract. The proseminal ghrelin, I think, is a enzyme that dissolves the clot. So a short time after semen is ejaculated, it forms a clot initially, and then this clot is dissolved. I think it's proseminal ghrelin that does this, that dissolves the clot, and it becomes liquid again. And then the sperm can move up into the fallopian tubes, up through the cervix and into the uterus and into the fallopian tubes. Okay, male sexual response. Remember, sexual intercourse, the copulation, coitus has got lots of names to it. We're going to stick with coitus is the official name of sexual intercourse. Circulation to penis is sympathetic. Remember, it's the, well, that's true for all circulation. Remember, arterioles are innervated by the sympathetic nervous system. Okay, the phases of male sexual response. Okay, excitement, vasoconstriction, myotonia. So obviously the penile arteries dilate. The erectile tissue is filled up with blood during male sexual cycle, and erection occurs. And then that's actually vasoconstriction. Myotonia is movement of stiffening of muscles, increases the heart rate, blood pressure, respiration, bulbourethral gland secretions. Erection. Parasympathetic fibers cause release of NO, nitric oxide. This is a gas, and it's also a neurotransmitter. And people were really surprised when it turned out such a simple molecule could be a neurotransmitter. And it relaxes penile arteries in the lacuna of erectile tissue. So when it relaxes penile arteries, lots of blood comes in. This is how nitrates work for people with angina. They take nitroglycerin. People didn't know how that worked, but it works by stimulating NO, nitric oxide, which causes vasodilation. That's why also Viagra prevents the breakdown of nitric oxide. So this is why men who have who take nitroglycerin are told not to take Viagra, because then you have two things that open up arteries, and not just penile arteries, but arteries all over your body, and this will cause your blood pressure to drop precipitously. The bulbospongiosus and issue cavernosus muscles, they are getting ready to squeeze the urethra to cause ejaculation. Intromission is just another word for putting a penis into a vagina, is intromission. And then of course we have penile thrusting, we have plateau. So you get this excitement, and they hit a plateau, and then those variables continue, and then all of a sudden a man gets close to orgasm, and you have two parts of ejaculation. Emission, sympathetic stimulates peristalsis and ductus deferens. So the peristalsis starts in ductus deferens. The prostate and seminal vehicle emit their secretions. Then expulsion, more secretions, internal sphincter for the urethra closes, so no urine can come out, you can't ejaculate and urinate at the same time. Somatic stimulation of muscles, compressed root of penis, compression of urethra, and sperm is expelled from the penis. So really, all three nervous systems are needed for male sexual function. First, you need the parasympathetic reflex to cause erection. You need sympathetic stimulation for peristalsis and ductus deferens, and then the muscles that compress the root of penis and compress the urethra, these ischiocavernosus and bulbospongiosus muscles, they're innervated by the somatic nervous system. So you need all three nervous systems to allow ejaculation. And then, after ejaculation, we have resolution. Constriction of penile arteries, contraction of trabecular muscles that compress the lacunae, detumescence, which is loss of the erection, cardiovascular and respiration returning normal, and then we have what's called a refractory period. So you hit climax, ejaculation, and then you go down to resolution, and you have a refractory period where you're not going to be able to do this again real soon. It could be pretty soon on a different magnitude, longer other, but there's going to be some kind of refractory period. We can't immediately do it again. And on that note, we're done with males. So I'm going to call it here, and then we'll pick it up on Repro 3 with females. Okay, see you.